almost all the way through immunology. I realize this is a difficult course, and we try to make it understandable and so that you can apply the same lessons in a variety of different situations. Now, one of those situations is a problem that's been plaguing the world for close to 40 years, 35-ish years now, and that is the HIV AIDS epidemic. So HIV is the virus that causes acquired immunodeficiency syndrome. And one of the things that's a problem with this virus is that it attacks the very cells that you rely on to control viruses. And so other viruses we now know do this as well, and they also are a problem for developing vaccines and for treating patients. So the first thing is, how big of a problem is this? And these numbers are a little bit old, but they've held fairly steady for the past several years. And so if you look at the actual numbers of estimates, you can see that in every continent, in every geographic location, there are a number of people infected in the millions, but the vast majority of them are in sub-Saharan Africa. There's only almost 25 million, or actually it's a little over 25 million now. And so the problem is that if we compare that to, for example, the ongoing coronavirus epidemic, there's many more people infected with HIV. And so this is still an ongoing issue. And right now, even today, even with all of the programs in place, there's still about 7,500 people infected with HIV every day. And about 5,000 people, particularly in these heavily impacted areas, including sub-Saharan Africa, are dying from HIV each day. Now, in just the United States, we've passed 4 million or 1 million infections with COVID-19, and we have about 1,000 people dying per day in the United States alone. So you can see that they are both still ongoing epidemics that really need solutions. So the question is, why is this such a problem? Okay, so many of you have probably taken the HIV course, but really, if we go back through the history of HIV, it was really discovered in the early 80s. And so the first it was actually discovered by two different groups independently. So Luc Montagnier and Francois Barsunosi independently isolated a oncogenic retrovirus from AIDS patients. And these are patients that had abnormal immune responses, they had deficient CD4 numbers, and they called this lymphadenopathy associated virus, or LAV. At the same time, Robert Gallo and Jay Levy, Robert Gallo in particular is a very interesting character, independently characterized human T lymphotropic virus, and as well as an AIDS-associated retrovirus. So you had a French group and an American group both isolating this, and perhaps prematurely, Gallo and Levy actually identified this as the causative agent of AIDS. It was not really backed by proof that it caused it, but it was always associated with these patients that had abnormal immunity. Now, eventually, they realized they were working on the same virus. When they published their results, they published sequences and they realized, oh, it's the same virus, okay? And so they renamed this human immunodeficiency virus, or HIV. Now, that was great, but at that time, we had pretty successful in making vaccines for a large number of viruses. So we employed our standard approaches, and I include myself in that group because I was part of the collaboration for AIDS vaccine discovery. And the truth is that all of those vaccine attempts were a complete failure. They showed no efficacy in preventing infection or disease. So the question is why? When you look at HIV, it's a retrovirus, and it has two genome strands in it that are identical. Okay, and so they're essentially a long RNA transcript that is as the proteins are being made, they're differentially hydrolyzed and cut up into smaller proteins. But there really is, the main ones are the GAG, Pol, and envelope proteins. GAG is the protein that organizes the genome inside of the virion. The envelope codes the GP160 protein that's cleaved into GP120 and GP41. So that's the receptor for the cellular ligand. The GAG protein here is in green, and that's really cut up and organizes the genome segments within the virus. And then the polymerase associates with, sorry, can't see it really on this one, but the polymerase is associated with the virus and allows it to be transcribed. Okay, so it has an RNA-dependent RNA polymerase. There are a number of accessory genes that have been identified, notably VIF, NEF, some of these other ones that are derived from intergenic sequences. And those are important for things that the virus does to evade the immune system. And they've been ascribed a lot of different functions. You don't have to know what they are, but they're really, excuse me, their job is to sort of disrupt cellular behavior so that the virus can keep proliferating. So when you look at the HIV life cycle, it's not that different from other viruses, right? Virtually all viruses follow these same steps. There's binding to the host proteins and then fusion. This is a little incorrect. It's fusion in endosomes triggered by the low pH. That dumps the genetic components into the cytoplasm of the cell. Now, one thing that retroviruses do that is a little different is that they reverse transcribe their RNA to DNA. And because they're including integrases in the viral genome, these allow that DNA to be inserted into the host DNA. That's one of the big problems, is that the virus can just basically hide out and not be noticed by the immune system. If those genes are not transcribed, nothing happens. But it's from those genomic DNA that then you get transcription of more host genomes and the proteins, translation of the proteins that then assemble the virus and release. So this is not very different from what we talked about for influenza, right? Where the virus is getting in, it replicates in the nucleus, it makes more of its own genome and more proteins, and then is released from the cell. So for that, it's really not, that on the surface doesn't appear to be the problem. The one problem is integration into the host DNA so it can hide out. But the bigger problem is what types of cells it infects. Okay, so here's just another way of looking at this, right? There's attachment. Now, one thing that's important for attachment is that it requires two proteins. It requires binding to the CD4. So the GP120, GP41 complex is going to bind to CD4, but then it needs a co-receptor. And typically, during initial infection, it's CCR5, but the virus can then mutate and change to allow for use of CXCR4. The main thing is that in order to establish a new infection, though, it really requires CCR5. Okay, so that's really to get things going. Now, once it binds, it fuses, it uncoats. There is reverse transcription, right? This is by the reverse transcriptase. That then is integrated into the genome. And you have these accessory proteins that modulate when the virus is actually going to transcribe and make new genomes. So if it gets transcribed, all right, some of these genes are made, and they then regulate expression of the genome. However, at some point, these cells become activated, and you get transcription of new genetic material as well as translation. And those genes typically are the ones that are important for assembly of the virus, and then there's release. So one key step there is for you to get new virus, it really requires cellular activation to overcome these sort of regulatory steps by the accessory proteins. So let's talk about how you establish an infection. It turns out that getting HIV uh, from from discordant couples, studies of discordant couples, it actually takes a large number of sex acts to transmit virus. So you have one partner with HIV and one partner without HIV. It requires about 100 sex acts to get an actual infection of the uninfected partner. But it actually matters a lot on how you're going to get it. Okay, so oral intercourse, it's 1 in 10,000 will give you an infection of HIV. It can happen, but it's extremely rare. When you're talking about vaginal intercourse, you really have to say if it's the donor. So in this case, if it's the male that has the HIV, then about 5 in 10,000 sex acts will actually transmit virus. If it's the female or the male that has HIV and the female, sorry, I spoke that incorrectly. If you're the male in vaginal intercourse, it's about 5 out of 10,000. If it's a female, it's about 10 out of 10,000. That's because there's just more damage to the female reproductive tract during vaginal intercourse. Now, in anal intercourse, again, it is whether you're the recipient or the insertive partner. And so if you're the insertive partner, again, it's fairly low because it's low damage to the foreskin as well as other areas. But there's more damage to the recipient, and so you get 50 out of 10,000. But still a fairly low amount. So even if you're talking about it's 5 out of 1,000 or 1 in several hundred, so basically a couple hundred sex acts to actually transmit virus. Now, one of the problems, both from a perspective of transmitting how much virus and the efficiency, is needle sharing between infected drug users. Okay, so in that case, exposure gives you a high transmission rate. It's actually a little bit higher than that even now. And so that's intentionally transferring blood from one person to the next. It's about 1 in 100 times. Now, what we do know from experiments from Danny Dewick at the NIH and others is that typically in everything except for IV drug sharing, in all of these, transmission is typically by a single virus particle. So one virus is getting in. And so that's the infection route. And again, that virus, in all of those cases, has to be CCR5 because those are the first cells that are going to be infective. The one issue is that during needle sharing, not only are you getting a higher rate of transmission, but you're transmitting more than a single virus. And so it could be CCR5 tropic or it can be CXCR4 tropic. And so you are already set up for failure, and those patients typically progress much faster because they're getting a much more diverse viral infection with a higher viral load. So the question is, in all of these, how is the virus getting to CD4 positive, CCR5 positive target cells so it can infect them? And it turns out that the initial infection is mostly through macrophages and other cells that are just under the epithelium. So if this is the columnar epithelium, right, in mucosal surfaces, then it has to get through a single layer, and you can see that this is either by direct infection of the epithelium, which is sort of the least common event, can be transcytosis of virus. We know that happens in vitro, but the main way is that it's going to infect cells that are stretching across the epithelium. And you have a lot of these cells at the mucosal sites, that are really there. Their job is to be there to sense virus. And so the infection rates in mucosal surfaces, either in the vagina or in rectal tissues, is higher because the virus can infect target cells right away. Once it does that and gets past the epithelium, then it can infect any of the cells that are already there. Okay, so that's sort of the normal situation. If you're talking about skin, which is much thicker than mucosal surfaces, there has to be physical damage, right? Most of the epithelium is actually dead or drying epidermis that is unsupportive of virus replication. And so that's why the rates for insertive sex are much lower than for receptive. Okay, so there has to be some abrasion. And this happens during normal intercourse. There is some damage to the epithelium. And that also can occur in the mucosal sites. Okay, so now when you're looking at the blood, right, really you're looking at what kind of cells are there. So CCR5 is expressed on macrophages and T cells, but CXCR4 is mainly expressed on T cells alone. Okay, so these are two looking at T cells in both the blood and the gut. And you can see that in the mucosa, most of the cells are going to be positive for CCR5 as well as CXCR4. But once the virus gets in, all of the T cells that are floating around are mainly CXCR4. And so you can see that you really are, if the virus is going to survive, it has to change to that receptor. Okay, so infection via mucosal routes is highly biased to CCR5 tropic strains, because that's where you see CCR5, is in the macrophages and T cells in the mucosa. And mostly it's the macrophages. And so you don't really have that in the blood. If you're talking about IV blood transfer, so you're talking about needle sharing, you can get either virus. And so the virus can certainly infect mucosal sites, but it's already mutated so that it can infect all these CCR5 negative T cells in the blood. Okay, so that's why initial virus infection is almost always CCR5 tropic, but during infection, the virus is mutating and changes to be able to use CXCR4 as the co-receptor. And that then allows it to spread rapidly through the T cells in the blood. Now, if you think about that, what that means, though, is that not all of the virus can change, because in order for the virus to get to the next individual, so we talked about viruses have to spread between hosts, they have to have some CCR5 tropic virus. And so this represents a bottleneck where the virus is limited in how much it can change, because in order to get to the next host, it has to retain some of that CCR5 tropism. So this change from CXCR5 tropic to CXCR4 tropic is not necessarily the main cause of disease, right? Because many patients retain CCR5 tropic virus, and they transmit it to others, and they progress to AIDS without changing to use CXCR4. So the question is, then why are they progressing? Well, I want to take sort of a side note here, or sidebar here, to talk about the Berlin patient. Now, he was the first. There's been a couple after him, and you can consider him either the luckiest guy or the unluckiest guy in the world. Okay, so he was diagnosed with HIV, and then he got a second diagnosis of leukemia. He actually had AML, or acute myelogenous leukemia. And so what happened is that then he was basically treated with chemotherapy and irradiation to wipe out all of his blood cells. And then that also wipes out the bone marrow, as we talked about. And then he got bone marrow transplant from a donor that lacked CCR5. Okay, so this is a mutation that's actually fairly common in the population. And so the virus could not infect the virus that he had could not infect any of the new cells that arose in him because they didn't have CCR5. And so this actually cured both his HIV and his leukemia, and it was the first known intentional cured patient for HIV. And so you can see here, here's his viremia. Right after the bone marrow transplant, it kind of spikes because he becomes immunosuppressed. But then after establishment of T cells and his immune system, you can see there's no viremia, and his CD4 T cell counts are going back up, which means that he's no longer classified as an AIDS patient. There's been, as I said, there's been a couple of others who had the same thing, and this has become one of the approaches to curing HIV is gene therapy. The drawback to this, of course, is that bone marrow transplants, as we talked about, can be quite devastating. They can kill the patient due to graft versus host disease, and as well as they're very expensive. Typically a bone marrow transplant in the United States is about $1 million. So you're really talking about a very expensive procedure that is not widely applicable to most of the patients who have HIV and AIDS, including those in sub-Saharan Africa.
Dr. Blattman?
Yes.
How could the donor be CCR5 negative? How is that possible?
So there are I forget the percentage, it's something like 0.1% of all people on the planet have a mutation in CCR5. It doesn't appear to cause any differences in your ability to make an immune response because you have other chemokine receptors that can compensate for it. But because this virus needs CCR5, it can't get in. So it's actually a common mutation. If we really look at all humans, we all have some mutation in some proteins. Okay, there's no way around it. We all are mutants, and we don't. If we look at just small nucleotide polymorphisms or non-synonymous mutations in genes, we all have some. And so this is actually a common one.
Thank you.
Okay, so let's get back to why does HIV then cause disease? Well, if we remember, viruses have to spread from the site of infection, they then go through the lymphatics to a lymph node, and then they spread throughout the body because eventually they drain back into the thoracic duct and are able to gain access to everywhere else. Now, the sneaky thing about HIV is that it's going to a lymph node where all of your CD4 T cells and your macrophages and other cells are. It's a rich source of cells that it can infect. So instead of just using this as a truck stop on the way to another destination, it's actually going right to the site where all the cells it needs to infect are going. And so it's able to infect those cells. And then those cells then, as they travel, right, those T cells, or they remain naive T cells, they're going to go through the blood and spread to all of the other lymph nodes. And so the virus disseminates very rapidly throughout the lymphatic system. Now, ultimately, those T cells are going to then replicate HIV, and that's going to be expressed in the mucosal surfaces, and it gets out essentially the same way that it got in. So it's going to be able to infect cells that are bridging that epithelial junctions and then transmit virus to others. So the question is, when you get HIV, do you feel sick? We all know about AIDS and the long-term effects, but when you first get HIV, you get a flu-like disease. It's rarely life-threatening, but right after, in 10 to 14 days after infection, you'll feel things like fever, right? You'll have malaise, which is general uneasiness, headaches. You can have your lymph nodes will swell up because you're making an immune response to it. You can have rashes, nausea, vomiting. So right after exposure, 10 to 14 days after exposure, you're experiencing a flu-like disease, but then it goes away. Well, that's one of the tricks or one of the problems early in diagnosing HIV was that, well, okay, the disease went away, but the infection didn't go away. And that's because the virus can integrate into the host genome and sort of hide it out. So during HIV infection, okay, you probably have all seen some version of this graph. You have some exposure event here at day 0. And what happens is the virus is going to replicate, okay, and then it goes way down. It's mostly, for the most part, it's controlled by the immune system. It really is just those few cells that have the virus integrated into their genomes that it's hiding out that are persisting. Now, on the other side, if you look at the CD4 count, right, in blue, the CD4 cells, initially the virus is going to infect those cells and they're going to cause lymphadenopathy, and they are mostly going to go away. So that's the first big problem. It's very difficult to establish that primary response to HIV because you need CD4 cells. And so if you have a large infection, you destroy your CD4 cells, that impacts everything that comes after. It impacts the antibody response, it impacts the CD8 T cell response. So we talked about without CD4 cells, the CD8 response is short-lived, it dies off, and so you don't have long-term control. The antibody response is not appropriate because you don't have somatic hypermutation, affinity maturation, you don't have isotype switching to the best isotypes. You also don't generate memory B cells as well, or plasma cells. So this initial drop in CD4 cells is the first big problem. What happens is then you establish some level of immunity, and that's called the set point. And we do know that the higher the set point, the better off you are, meaning the better immune response you generated initially, the slower you're going to progress. If you have a very low set point, or in terms of if you have a very low set point of virus and a high set point for the T cells and immune response, you're going to do pretty well. If you have a very high set point for virus and a low set point for immune response, you're going to progress much faster. Eventually, what happens though, over many years, is that eventually the virus keeps surging. It can be controlled, and this would be analogous to the sort of equilibrium phase in cancer, where the immune system is battling it, the virus is mutating, and eventually the virus is going to mutate and win. And that's when you start to see symptoms, including drops in CD4 count. Typically, if it's below 200, that is indicative of an AIDS patient. And then once that drops, then you have the problem with all the other opportunistic infections that eventually you're not going to die from HIV, you're going to die from something else that gets in you and infects you that you can't control. Okay, so what happens that initial immune response turns out to be the critical part in predicting how fast a patient's going to progress and what you can do about it. Well, when you think about it, this is the exact same response that you have every other time, right? You have antigens being delivered, so virus, killed virus, and is coated with complement proteins, including IC3B. It's going to be displayed by follicular dendritic cells. That stimulates B cell response, okay? That's the first step. Then you also have T cells. And those are going to be initiated by antigen presenting cells. So you have both the CD8 cells that are being activated on the antigen presenting cell and the CD4 helper cells that are being activated. And those are the critical ones for helping B cells and CD8 cells. So these helper cells for CD8 T cells, they're mainly making IL-2 that will support their growth and long-term survival. But they're also making things like interferon gamma, IL-12, that are biasing this towards a Th1 response. Th1 responses are good for systemic viral infections, and that's what you want. Okay, so it's making Th1 cytokines and as well as IL-2 to support it. On the other hand, for B cells, it's providing that key CD40 ligand signal to the B cell that then tells that B cell, okay, you need to go out and make, you need to form germinal centers, make higher affinity antibodies, and then ultimately generate memory B cells and plasma cells. That is not any different than anything else we've talked about this semester. This is what you do for every viral infection. The problem is that the virus gets to infect some cells and hide out. And the main cells it's going to infect are these ones. And so that's the problem. If you lose that response during the initial infection, everything else kind of goes in the tank. Okay, so what are the problems? So if you don't have this good CD4 support for B cells, you have a problem with initially somatic hypermutation and affinity maturation. And part of the problem is you need extensive mutations to actually trigger neutralizing epitopes. Now, it's sort of a misnomer. If you think about it, every HIV patient eventually develops some neutralizing antibodies to their own virus. This idea that you need not broadly neutralizing antibodies is a vaccine construct. For a vaccine, you have to have broadly neutralizing antibodies to all viruses, and that's a much more difficult project. But when you get antibodies to HIV, if they're not highly mutated, most of them are just basically going to see this glycan shield on the envelope proteins. Okay, so here's GP41. It's not drawn here, but it's the stalk that sticks through the membrane of the virus. And then GP120, which is this part here, is the part that's going to recognize CD4 and the co-receptor. But targeting that glycan shield doesn't neutralize the virus. It's a big problem is that most viruses initially are not neutralizing. And so in order to do that, to get antibodies that are neutralizing, you have to target parts of the protein that are exposed that really are either going to the part of the GP120 that recognizes CD4, so it prevents binding, or there are antibodies that will recognize parts of GP41 called the membrane proximal region that will prevent the envelope protein from folding open and allowing fusion. Okay, so you either have to prevent binding or prevent fusion to allow the virus to dump the contents into the cell. So neutralizing antibodies usually develop later infection, where it's too late to prevent disease progression because you've already lost your CD4 cells. That's why that initial response is so important. Now, neutralizing antibodies can really help, but their job is mainly in the initial steps, right? If they're helping out, they're doing it early. After the virus is disseminated throughout the body, it's very difficult to neutralize all of it, including because it's integrated into the genome and it can hide out. Okay, so what happens then is even if you get these neutralizing antibodies, just like in tumor equilibrium, there's going to be mutation to escape from it. Okay, so tumors typically try to escape from T cells, but viruses will mutate and change their envelope protein to escape from the antibody response. So there's a lot of problems with generating a proper B cell response to HIV. But when you look at it, really, the antibody response is not very different from most other viruses, right? Here's the virus coming in, gets mostly controlled. That control is primarily by CD8 cells, okay? So you can see the peak of the virus control occurs, or most of the virus control coincides with the peak of the T cell response. On the other hand, the antibodies really are taking a longer time to kick in, primarily, right, because the CD4 cells get impaired. That initial dip is a big problem for the CD4 response. Okay, so this again is coming back to that initial response. The better that is, the better off you're going to do. Okay, so let's talk about the CD8 response. That's really what you need for virus control. Antibodies can help, but really to control viruses, you need a strong CD8 response. And so what happens is you have activation of both CD8 cells and CD4 cells on your dendritic cells. And those then are going to differentiate into effector cells and be able to go out and do the things they need to do. The key here is that CD4 cells are making things like IL-2 that support the effector response. And I would say this is a figure that from somebody who sort of got things mixed up. There's not a memory cell component here that's actually going down here to memory cells. It's a direct line to the effector CD8 cells here. But in any case, what's happening is these cells will go out and they're, first of all, CD8 cells are killer cells. That's their main job. And so they have perforin, granzyme. They can use the fast ligand pathway. They can use, and so that's causing apoptosis of infected cells. They're also making cytokines, for example, TNF, that can shut down in cells or in some cases kill them. And it primes them to respond. It makes them ready to go. If they should get infected, they know to shut down. It'll also make proteins that these are your normal chemokine receptor proteins, and they will block binding of primarily binding of the virus to the co-receptor. So if the chemokines bound to the co-receptor, the virus can't bind. Okay, so that you need this response, and the better that response is, the better off you're going to control it. And that all comes back to the helper cells. If you have a low infection and good helper response, the effector CD8 response is good, then you have better long-term control. And so it really comes back to this equilibrium concept, right? And this was initially thought of for HIV, and then Bob Schreiber and others developed this for tumors, in that there's an acute response. And from that, you get, the stronger that is, then the better you are to control HIV. And so this is showing HIV viral levels. And so you can see if you have a very strong CD8 response, again, coming back to a good CD4 help for that response, then you get slow progression. If you have a weak T cell response, then you'd see that the viral set point is much higher, and so you get much more rapid progression. And so typically what that says is that here, if you have one virus particle, right, then it takes a little bit longer for the virus to replicate, and so you get typically a stronger response. IV drug users up here get transmitted of a lot of virus. It spreads very quickly, and you get a worse CD4 help, and so you get a very weak CTL response, and so they progress much faster. But eventually what's happening is that, yes, that set point will delay progression, but all groups progress to disease, okay? So whether it's slow or rapid, it's going to eventually happen. And so that has to do with the deterioration of the CD4 T cells. Those are where the virus is hiding out. So when you eventually lose those, if you lose enough CD4 T cells, you can't make good responses to other things. And so that's really when disease sets in. And so that part of that has to do with, okay, you can't make new responses, but you also can't support the existing responses. So one of the reasons that the CTL response fails is that that was one of the things that we talked about. CD8 T cells, they proliferate, expand, they go do their job, but they're not designed to keep going forever. And so what happens is there's more viruses there. In this case, it's called antigen load. You're actually getting more stimulation of the T cells. And the more stimulation in a chronic setting, the CD8 T cells will start to lose function. Okay, so if you have more antigen or less CD4 help, then you get, first of all, expression of things like PD-1. We've talked about that. That's one of those genes that shuts off T cells. So when you look at the more virus and less CD4 help, you go from a cell that can make cytokines, it can kill, it can proliferate, it survive, and then it starts shutting things off. And it does this in a sequential manner. The first thing is the response can't keep going up. And so it'll shut off IL-2 production as well as proliferation. It'll also shut off immediate killing. They can still have some function, they can make some cytokines, but they're suddenly going to be impaired in their function. And as that progresses, then you can see they lose cytokine production. They're not able to make IL-2 or the IL-2 receptor, so that impinges on their ability to proliferate. They can't survive, and eventually they'll die off. So they'll undergo apoptosis. Okay, so again, coming back to the idea that the more virus that's there results in worse CD4 help. As that CD4 help goes away, you can't support your CD8 T cells, so you can't kill the virus. So what happens here? Well, Julie McElrath's group in Seattle, as well as others, have looked at this and said, well, what happens in this sort of equilibrium phase? And you can see that here the the solid line is the virus, and the purple and the dotted line, the solid line is the virus. The dotted line is actually the immune response to that virus. You can see there, here's our initial virus in black, okay? And it infects. You get a CD8 response to it, and virtually all of it goes away. But just like in cancer, right, you can kill 99% of it, but you're selecting for resistant strains. And so you get a mutated version of HIV, and that goes up. You get an immune response to that, and that is controlled. It mutates more, so you get the blue line. There's an immune response to that, and that's getting controlled. And the green one, and the teal one, but eventually what happens is that the virus, in addition to causing exhaustion, is escaping. So eventually it's going to mutate epitopes that allow the virus to not have anything that's going to be seen by T cells. The virus can go up, but there's nothing that can see it or control it. So there's a downside to that. Okay, so the downside is if a virus keeps mutating, it becomes less fit, or it's less able to replicate. And so the downside to the virus mutating to get away from the CD8 response is that now it can't mutate very well. That doesn't make a great outcome for it being transmitted to the next host. And we know this from associations with people who are resistant to HIV infection or have lower viremia, have particularly MHC, that if the virus mutates that epitope, there's a big fitness cost, and so those people are typically resistant. And so and people who have HLA-B51, right, will select for a variant, this I135X variant, that you can see that certainly can present those epitopes and select for that variant. That's what the virus has to mutate to escape from the T cells. But if you see, the mutant doesn't grow very well. Okay, so selection of epitope escape viruses has a replication disadvantage for HIV.
Dr. Blattman?
Yes.
Why don't they just treat HIV patients with an anti-CCR5 antibody if you can survive without that protein?
Well, if we go back to the passive immunization, right? What does it cost to treat somebody for a single scorpion sting?
It would cost a lot of money.
$80,000, and you would have to keep doing it, weekly. And so it's not a long-term solution. And then on the other hand, you can't really transport antibodies to sub-Saharan Africa and try and treat people that way. People have tried and thought of that, but it just doesn't work very well because you have to give such high levels of antibodies to coat all of the CCR5 positive cells that it just wouldn't work very well. The virus is going to find a way around that.
Gotcha, thank you.
So when we look at protective versus permissive HLA alleles, and I encourage everybody to go and find out what their HLA alleles are. But if you look at people with B57 or AO2, right, that you can have typically they have lower viral load. Okay, so again, lower viral load means you progress slower. Where there are individuals that have high or permissive alleles, B45, B18 are some of the more common ones, they have typically higher than average viral loads. And so this is where we begin to understand that if you have an MHC, that if there's a high fitness cost for the virus mutating the epitope that's being presented there, then the virus can't replicate as well. And so that would be the protective alleles. If you have an MHC that presents an epitope and the virus can mutate that with virtually no fitness cost, you can see that it has higher than average replication. And those are the permissive alleles. But it doesn't really tell us why you progress to AIDS, right? And that takes, a long time, 10 years or more in some individuals. Well, you have to really go back and look at what are the different steps in the immune response. Okay, so we have the virus coming in here. It replicates. This is on a log scale, but it's going down depending on what your set point is. It goes up and it comes down. Well, you sort of correlate that with when are the different parts of it happening. So the CD8 response is occurring. That causes the virus to start going down. Okay, so that's good. That's what you want. The better that CD8 response depends on the better CD4 response, and so you're starting to get that. And you're getting some initial antibodies. But those don't really help you. Really, it's you get a CD8 response, and so you're talking about T cell exhaustion, or the virus is just going to escape. Okay, so there's this constant battle or equilibrium phase where it's the virus is mutating, T cells are trying to control it. Eventually, the virus is going to win. It's also going to escape from neutralizing antibody. Eventually, every individual will develop autologous virus-specific neutralizing antibody, meaning make antibodies against the virus they have, and it can neutralize it. But then the virus is going to say, well, I'm just going to escape from the antibody response. Now, when you lose those two things, essentially the virus is being able to go anywhere it wants and do whatever it pleases. And so that's when you get this infection. There's a next step, basically, is the loss of CD4 cells. Once the virus has escaped from CD8 cells and from antibodies, there's nothing to prevent it from infecting CD4 cells and causing them to be destroyed. And what happens here is that initially, when HIV is going to infect cells, initially it's going to infect resting CD4 cells. Okay, so they're just CD4 cells that are hanging out in the lymph node. The virus traffics there, and the main result of that is integration into the host DNA, and it's not producing any new virus. We don't know all of the factors here, right, that are controlling this. We do know some that are preventing nucleotides from being incorporated, but essentially what's happening is that's your reservoir. And so as the virus is hiding out, once in a while it'll get activated, but again, what is the frequency of activation of CD4 cells? So it's 1 in a million antigen specific. And so over time, after you've lost CD8 control and antibody, the virus is in these cells and that cell gets activated. And once it does that, right, then it's reactivated from the genome and you get virus produced. So it's really in the activated CD4 cells. So if you think about this long term, what's happening? Somebody's infected with HIV. They develop CD8 responses and B cell responses. Eventually those get kind of escaped from, but you're in a dirty world, right? You're getting flu infections and rhinovirus infections, common cold, getting adenovirus infections, you're getting bacterial infections. Every time that happens, you get a burst of HIV because it's activating antigen-specific cells for those other pathogens. So over time, you're losing these cells, okay? You're essentially, the virus is making a big burst, it makes more virus, it infects all of the other naive cells, and eventually all of your naive cells are infected. And so over time, you're losing that population. So how does it get activated? Well, rarely it'll be because of recognizing HIV-specific antigens. It can happen, right? But it's not usually that. It's, you get much more exposure to other environmental things, other pathogens. So you can have naive CD4 cells are activated against influenza. That will reactivate HIV from those cells. You can also have infection, if you think about it, naive cells can also be develop into Tregs. And so if you have Tregs getting activated, then it's really a reaction to self-reactive antigens, okay? So autoantigens contribute to that. Over time, you have sort of homeostatic mechanisms that are inducing pseudo-activation of naive T cells. So those are helping to, those are sort of homeostatic mechanisms that just maintain your T cell levels. And so there's some slower tick over of those. But the main mechanism is that you are constantly making responses against things in your gut mucosa. Okay, this is a normal part of every day. If you've had eggs for breakfast, you're making an immune response to those right now. It's not an inflammatory response, but HIV doesn't need an inflammatory response. It just needs you to make a response. So you're really talking about this response to gut mucosa. One of the issues, though, is that we know that if there's breaks in the gut mucosa, if you have some damage there, you get some microbes will cross the epithelium, and that'll expose T cells to toll-like receptors, things like LPS. We already know that strong toll-like receptor signals can non-specifically activate CD4 cells. And so Jason Brenchley, who again was in Danny Dewick's lab, proposed this model. And it isn't a model, it's not it's not fully accepted. It's one of the mechanisms that can contribute to AIDS. But I think it's one that sort of explains the progression to AIDS the most. And it's really as you have infection of CD4 cells, you're losing immunity at mucosal surfaces. And what's happening then, if you lose immunity, more things are crossing the mucosal epithelium, and that then lends to more activation of CD4 cells, more loss of immunity at those surfaces, and that's ultimately where you progress to AIDS. And the way that he looked at this was just looking at plasma LPS levels. So he had uninfected, healthy individuals, people with acute HIV, early infection, people in chronic infection, and then those who had progressed to AIDS. And you can see that as you progress to AIDS, you have more LPS in your blood. Well, you shouldn't have LPS in your blood, right? As you can see that those patients are progressing faster, and the idea is that they've lost mucosal integrity, so those CD4 cells keep getting activated by strong toll-like receptor signals, and that then leads to them proliferating and making more virus. Does it explain everything? No, because look, these people are progressing to AIDS. They don't have high toll-like receptors or high plasma LPS. So it's not the only mechanism, but it is certainly one of the ways that contributes to progression to AIDS. But ultimately what's happening then is you lose CD4 T cells, okay? That is the defining characteristic of HIV-induced AIDS. So some of the things that can happen are that that HIV can cause stimulus of cells in the thymus to actually delete HIV-specific cells. But the primary way is that you have export of naive cells from the thymus. Those go out into the periphery, And those are then being, every time a different CD4 is getting activated, the virus is going to spread to those other cells and infect them. Over time, that can cause basically all of your CD4 T cells to be infected. Now, the mechanisms of this, of why the cells disappear, are not completely understood. Okay, so we know that some HIV genes can induce apoptosis in infected mature CD4 T cells. That's not a great thing for the virus to do, because why would it want to kill off the cell that's making more virus? So that can happen mostly in cell culture. So the role of that in vivo is not really known. Again, one of the things that can happen is those CD4 T cells, if they're exposed to things like LPS from disrupted mucosal epithelia, those cells can become activated to produce more virus. And it can stimulate them to undergo apoptosis on their own. But the main mechanism, once those cells are making virus, right? Once the cells are making more HIV proteins, then those CD4 cells are just like any other cell. They present it on their surface and they get killed by the CD8 T cells. Okay, so there is killing by the CD8 T cells, but the problem is it's hard to explain that, because late in the response, you've got exhaustion and escape from CD8 T cells. So it's not clear if there's intrinsic virus killing or if it's the immune system. What most people think is that as the virus is going along, the CD8 T cells retain enough recognition that they will kill off the CD4 cells. Okay, so that's sort of the pathogenesis of it, but a lot of the problem with HIV and AIDS was defining AIDS. And that seems like it would be an easy thing to do, but remember, you're not dying from HIV, you're dying from something else, either cancer or a other infection that normally wouldn't be lethal. And so the definition of AIDS really has three things. You have to have HIV positive. You have to have a CD4 count that's less than 200. So that's, down in the bottom 14% of people. So that's really low CD4s. And then you have to be diagnosed with something else. Okay. If you have just HIV and CD4 counts are low, you don't have anything else, there's no disease. So definition of disease includes those three things: HIV positive, low CD4 count, and then something else. And some of these, like Kaposi sarcoma, really only occurs in a subset of Middle Eastern men. You don't see it anywhere else, but you see it also in AIDS patients that are not genetically related to those Middle Eastern men. You can see there's a lot of opportunistic infections, right? Coccidiomycosis, which is common in the valley here. You can have things like toxoplasma. You can have recurrent bacterial infections. You can have tumors, including Burkitt's lymphoma or immunoblastic lymphoma. But a lot of things like cytomegalovirus, most of us have that. But reactivation of cytomegalovirus is very uncommon if you're not an AIDS patient or if you don't have HIV. Okay, so that was really the problem. And there are still faculty, notably Duesberg at UCLA, who keeps trying to say that HIV has nothing to do with AIDS, that it's more due to inhalers and things, and that's just completely been refuted. But there are people who still argue about the definition of AIDS. Okay, so that's really what happens. Initially you have the immune system making a response. The better that response is, the slower you progress. But over time, all of those naive CD4 cells get activated to produce more virus, and eventually you're going to lose the CD4 cells. When that happens, then you're going to get opportunistic infections or cancers that then are going to cause the actual disease and eventually death. Okay, so that's really the, in a nutshell, progression of AIDS. So what do we do about it? Well, the first thing in Western countries or Western civilization countries, we have a number of antiviral drugs. And believe it or not, we don't have antiviral drugs for most viruses. There's a few, but they're very limited and they're sort of just general things. We have many for HIV, but they were all developed because of this global epidemic. So there are things that will block fusion so the virus can't get in. There are those that block binding, just like the antibodies. There are some that will block uh, integration or replication to make DNA. There are those that will block the integration. And then there are some that will block maturation of the virus and sort of getting out. All of those work. In the early stages, though, what would happen is that doctors would treat with a single, you'd be treated with one drug, and once that would fail, you'd be treated with a second drug. And once that failed, you'd be treated with a third drug. And eventually you'd run out of drugs, and the virus you would progress. We now know that a better strategy is treat with several drugs at once. It's much harder for the virus to escape and make that and become resistant to that drug if it's also got to mutate and escape from more than one other drug. Okay, so now people are treating with multiple drugs at the same time. And that's shown here, right? So initially, here's your virus load. You treat with highly active antiretroviral therapy. The virus goes down, but it's not going to zero, right? Because you have this reservoir, and every time those CD4s are activated, the virus can mutate. And eventually you're going to get something that's resistant to that drug, so the virus will rebound. Now, many times, just like escaping from the immune response, you'll get a fitness cost to the virus if it's resistant. Any mutant is likely to have a fitness cost. And so here it comes back to a lower level, but it's still replicating. But if you start with many drugs here, instead of just a single one, then it's harder for the virus to mutate multiple sites at the same time. And so you get typically long-term control. And you can look at people like Magic Johnson, who've been living with it for 25 years now, and he's got no disease. So drugs can control this. Multiple drugs can control this for long periods of time, presumably for the rest of your lifetime, but they're not available to most people in the world, including those in sub-Saharan Africa. And they're hard to distribute there, and they're very costly. So it's not a solution for everybody, but it does work fairly well in Western countries that have access to this. So that brings up, well, what would be the best way to prevent HIV? And the answer to that is, of course, a vaccine would be the best solution. Okay, and so most vaccines to date have been abysmal failures. And this one is from 2000. These results are from 2006, 2007. And this was a vaccine from Merck. And it was an adenoviral-based vaccine, so recombinant adenovirus expressing HIV proteins. And it was given to thousands of individuals. And so then what they did is they looked at the infection rate in those individuals. And so you can see the black line here are people who were given the placebo vaccine. The red line are those that were given the actual adenovirus vaccine. Well, what happened is everybody made a response to the virus. They could detect anti-HIV T cell and B cell responses in greater than 90% of everybody they vaccinated. But what they found out is that infection rates in the vaccine group were higher. And so it's actually an example of a vaccine that made things worse, right? You actually increased your risks of becoming infected with HIV. So this This vaccine was canceled at an intermittent evaluation phase. So I'm sure you've all heard of the coronavirus. The vaccines, they took an initial look to look at efficacy, if they should keep going. Looks like yes, they should for that. In this case, they looked and said, no, it's failed already. We know that it doesn't work. The only successful or partially successful vaccine trial is the RV144 HIV vaccine trial that was conducted in Thailand. And this required a monumental effort. First, it was carried out by the US military, and they vaccinated tens of thousands of individuals. Okay, so there's almost there's a little over 16,000 individuals in this vaccine trial. And they vaccinated people, but they did it with 2 vaccines that had already failed to show any efficacy alone. So they did a canary pox. This is a pox virus that infects birds. It doesn't infect humans. It can get into cells and produce proteins, but it can't replicate. So it's actually a pretty weak vaccine. And then they had a protein boost. Both of these had been in their own clinical trials. They had both completely failed, but they had them in the freezer. So they said, okay, well, let's combine them. Maybe it'll work together, which is a dubious scientific assumption, but that's what they did. Now, what they found was that in the vaccine group, there was slightly lower infection rates. There was about 30% lower. But the main difference is really this initial part. Okay, so long term, this curve or this line have the exact same slope. So it doesn't provide long-term protection. It really, what most people think now, is it affected early events soon right after vaccination that then cause those individuals to have lower infection rates right off the bat. Why? We don't know. There was minimal to no detectable T cell response in any of the vaccines. The antibody response was detectable, but it's not broadly neutralizing. So how did they get this positive result? Well, first of all, they had to mess around with the statistics. If you notice the title of this figure, this is taken from the actual paper. This is the modified intention to treat analysis, meaning this is what we intended to do. So they had to take out people who didn't get the full set of vaccines and include people who got some non-standard numbers here. So the statistics are a little questionable here. To reach statistical significance, a p-value of less than p04 really required some playing around with the numbers. So it's not a long-term solution, and most people think that this is some kind of adjuvant effect that is really changing the initial steps of infection. However, this is the only one that's shown any efficacy. If you can look at here, vaccine efficacy is about 31%. Okay, even given the modified statistical analysis, there's some efficacy. Now this is brought into sub-Saharan Africa as a vaccine, and it's ongoing clinical trials. Here's the problem. We talked about the risk of risk exposure or behavioral risk that leads to exposure. This Thai group was actually a very conservative group, and typically that you would categorize these people in the very low risk population, and so most of these are probably low exposure numbers. If you go to sub-Saharan Africa, the exposure numbers are much higher, and you're talking about a much more diverse viral population. In Thailand, it's a very narrow population, so you wouldn't necessarily need a broadly applicable vaccine. If you go to sub-Saharan Africa, where it's mostly clade C viruses, it's a much more diverse group, and it's going to be much harder to control. My prediction is it's going to fail terribly in sub-Saharan Africa. It's going to show no efficacy. But if I'm wrong, I will eat my words. I hope that I'm wrong. It's a better way to say it. Okay, so I'll end there and I'll take questions either about this lecture or any of the other questions you might have on the previous lectures.
I have a quick question about the HIV test. So, when we can say the good time for doing the test to see that if someone has the HIV or not?
Well, you can detect it up to as soon as like 12 hours after infection.
Mm-hmm.
But certainly within a day or two after infection, you can certainly detect virus. Or a day or two after exposure.
Mm-hmm. Good. Thank you.
Dr. Blattman?
Yes, I can barely hear you.
Oh, sorry. Do you know if there are any mRNA vaccines for HIV in clinical trials right now?
In clinical trials, no, I don't think there are. And again, if you're following the Moderna or other vaccines that are mRNA vaccines that are being developed, what's their big problem? They're easy to make, they're quick, right? You can. That's why Moderna and Pfizer are out in the lead. But on the other hand, the transport after you make them is the hard part. These require transport on liquid nitrogen or at the very least dry ice, but you're still going to get some degradation. RNA is unstable. So if you have an RNA vaccine, yeah, you could potentially use that in Western worlds, but most of the people living with HIV are in sub-Saharan Africa. So really you need a vaccine for them. People that would benefit, another way of saying that is people who would benefit from an RNA vaccine already have access to antivirals, so they don't necessarily need a vaccine. They could just do treatment. People who need a vaccine and don't have access to antivirals, it wouldn't work because you can't transport it there very easily.
Dr. Blattman,
Yes.
on slide 14, I think maybe I just heard you wrong, but you had said that if you have a high set point for the immune response and high set point for the virus, then you progress faster.
Say that again.
If you have a high immune response and high viral load, then you said that you progress faster, or maybe
No,
I
if
heard
you
you
have a
wrong.
high immune response and a low viral load, you progress much slower. If
Okay,
you have a low immune response and a high viral load, you progress much faster.
gotcha. All right, thank you.
Okay, so one of the questions is the idea of some people are estimating that HIV only affects a small percentage of CD4 cells, and that this loss of CD4 cells is due to massive cell signaling, even if they're not not infected. That can happen. Toll-like receptor signaling on its own, in the Brenchley model, toll-like receptor on its own can actually kill off CD4 cells. It activates them and then they die off. That's part of it, but also every time those CD4 cells get activated, they're making new virus that infects more CD4 cells. And so there are a number of things happening. I think the 1% of that you're talking about, the low percentage of CD4 cells that are infected, that's not at the end. When you're actively undergoing AIDS, it's actually because it multiplies exponentially. That 1% becomes a large percent as you progress right at the end of AIDS. During that long equilibrium phase, you're correct. It's a very low percentage. But over time, it sort of creeps up, and then eventually right at the end, it's exploding. Okay, any other questions? All right, if there's no other questions, we'll see you next Tuesday. Have a happy Thanksgiving.
Thank you, you too.